Good morning, everyone. Good morning. Let me offer my welcome to everyone to the 2019 Marchase Endowed Lecture. My name is Chris Brown. I'm the Vice President for Research here at UAB. I started here almost exactly 2.5 years ago and followed in the footsteps, very large footsteps, and stand on the very broad shoulders of Dick Marchase. He was here, and in addition to being the Vice President for Research and Economic Development, he served here in the role of Professor, Department Chair, Associate Dean, and Interim President. So I guess that means he couldn't hold a job. But he's in retirement now, and so as far as I know, he's doing very well at retirement. Throughout his career, Dick was instrumental in and supportive of interdisciplinary research, which is why we're here today. His legacy continues to be felt by faculty-to-faculty interactions, groups of faculty coming together at this university and beyond, and especially through our university-wide Interdisciplinary Research Center, UWork program, which exists today and is doing very well. So in honor of his long service to the university, gifts were given upon his retirement for the University of Alabama at Birmingham Friends of Richard B. Marchase, PhD, UWork Endowed Award Fund, which I think wins the contest for the longest fund. And we use those funds to support this lecture series, of which this is the second one, and an award for a UAB investigator who leads and promotes interdisciplinary research across campus in the spirit of camaraderie that Dr. Marchase personified and personifies. The Marchase Award winner will be selected later this summer, and we'll have an award ceremony and a lecture at that time for the award winner. So it's now my pleasure to invite Dr. Selwyn Vickers, our Senior Vice President for Medicine and Dean of the UAB School of Medicine, to introduce our speaker for the 2019 Marchase Endowed Lecture. Selwyn.
Thank you, Chris. And Dick, it's an honor to have you back, and it's an honor to have this recognition for you annually, and the legacy of what you've contributed to our university. Our medical sciences and research will last, I think, in perpetuity. So thank you, and thank you, Gail, for being here as well. This is an honor for us to have Dr. Tay back. It's been, as I just asked him, it's been about 15 years since he's had a chance to visit UAB, and his comment, there have been a lot of cranes, and there is. We're sort of a different place than we were 15 years ago. But I met Dr. Tabak probably in 2009 as he was in a role of acting interim director. And I would say as much as anything, he's a consummate scientist and an outstanding administrative leader. His role before getting to NIH was Senior Associate Dean of Research and Professor of Dentistry and Biochemistry and Biophysics at the University of Rochester School of Medicine and Dentistry. And there he grew an outstanding program looking at glycoprotein biosynthesis, and over time earning him what really is, and certainly back then, a tremendously prestigious award that were given to few people, a merit recipient. And as you know, an extended level of funding, most of all demonstrating a high level of confidence in his science by his peers to allow him to pursue his science. In 2000, he became the director of the National Institute of Dental and Craniofacial Research, and he served in that role for 10 years. And since then, he's moved on to really, I would argue, to be the glue of the NIH and its trans-NIH initiatives. He, for Dr. Collins, has led a number of cross-cutting themes through the NIH Common Fund. He's led the task force on integrity. He oversees the error process for the extra money that was put in during that hiatus. He's also taken a keen interest in overseeing the Advisory Committee on Diversity in the workforce, as well as the overall workforce for science. And he's also led the task force for the trans-NIH study for environmental influences on child health outcomes. He continues to have a lab that's a part of the intramural NIH research program, and obviously is a fixture and a recognized national leader in his own right for his science, recognized also as he was a member of the Institute of Medicine, now called the National Academy of Medicine. So Dr. Tabak, we're glad you're here. It's been a long time, well overdue. We're excited and look forward to your talk and hopefully give you an outstanding day and visit at UAB.
Well, good morning, everybody. I want to first thank you for that overly generous introduction. So I'm going to be honest. Why am I here? I'm here because of this guy. That's what put it over the top. I was sharing with your chief of staff that the last 3 weeks I've actually taken 3 red eyes because I've been jetting around the country doing some formal consultation with tribal nations. And it's been an unusually busy time. But when I learned that there is a lectureship honoring this man, the answer had to be yes, okay? So as a former research dean, once a research dean, always a research dean, right? I mean, that's the way it works. This was the dean of research, right? All of us looked up to what Dick did. And so it is really my great privilege to be here, to say that and to honor you. So thank you. So what I'd like to do today is cover a few topics. Quick introduction, tell you a little bit about basic science, because we're always having to remind people about the value and importance of basic science. Speak to you about some of the research essentials that my colleagues and I have been engaged in partnership with all of you, in partnership with the extramural community. And then all of the faculty can leave, and if there are any trainees, you can stay, because I want to give you the secret of how to plan your path forward career-wise. But I don't want the faculty to see it, so they're all going to leave, and I'm only kidding, but okay. So by way of introduction, as I think you all know, NIH is the steward of medical and behavioral research for the country. Our mission statement's a very simple one: science in pursuit of fundamental knowledge about the nature and behavior of living systems. That means we have to do basic research. Very often, policymakers will say to me, well, why don't you just go right to the stuff that's going to, cure disease? What do you have to do all this basic stuff for? And it's somewhat humorous, but it's also a little scary, right? And so you really have to take the time to explain why everything is built upon that foundational work, that fundamental science. But that said, we are then obligated to apply that knowledge to extend healthy life and reduce illness and disability. So I think this is what it's all about, right? It's about the resource set that we are privileged to oversee to support research in this nation. And presently we enjoy a budget of $39 billion a year. About 11% of that is spent at NIH, and the rest is spent in the so-called extramural program. And that split 10 to 90 is, been roughly the same for many, many years. Now, if you drill down, you see that we fund scientists across the United States. The heat map shows the intensity of funding, and the darker the blue, the more the funding in a particular area. You don't have to squint to figure where Alabama is. Here you are. And Alabama has about 700 awards, most of them coming from this institution, and you can see the current numbers there. So we had a little fun with this to try and get a sense of what you're all famous for. And you should write down what you think the top 2 or 3 things are, and then let's see if it matches up with our analytics, okay? So we assessed the institution as a whole by looking at all of your publications from 2014 to 2016. So admittedly, we are out of date, and I'm sure it's much, much better now, but that's the frame that we used. And we looked at UAB, and then we looked at the whole rest of NIH, okay? And we used a series of metrics which are listed there, and then we compared which topics generated the most influence in their respective fields that your colleagues and all of you contribute to. And I won't go into the details, but just to show you that, in fact, if you look at over almost 3.3 million publications, and those that are supported by NIH and those that were derived from work here at UAB, you can see, and I'll just point you to the bar graphs, that UAB is doing really well, and in common with NIH-funded investigators, much better than those individuals who are doing research without NIH funding. Now, you can derive from that what you will, but that is what the data show. And what we wanted to figure out was whether, how is this productivity distributed? Do you have 100 areas of excellence where you're really making influence, or is it more focused? And in general, I will tell you, every institution I have ever visited, it's always focused. You can't be all things to all people, something that I'm sure Dick said to many of you multiple times over his career here. So this is a map of all the NIH-funded publications, all of NIH, and the size of the bubble gives you a sense of how influential the work is, NIH-funded versus other, and you can see certain specific areas. So protein structure I'm particularly interested in, so I always look at protein structure. But you can see things like population studies and epidemiology, the microbiome, cancer metastasis, GWAS studies, all of these different things. And when you now look at this for UAB, certain things pop out, which is very interesting. And I'd be very curious to know if this comports with what your view of your institution is. But in terms of influence, things that are sort of yellow to red are really leading the way in terms of their influence. Things that are green are more neutral in terms of the influence. And so clearly, epi is a hot spot for you. Clearly, work in premature birth, work related to hospitalization, cancer metastasis apparently is quite important here. And so a comparative effectiveness research, interventional research, these seem to be the areas that the sort of unbiased analytics say are at the forefront. Now, again, it'd be fascinating to hear what all you say about this, and so maybe when we meet later today, you can tell me this is a pile of stuff, or hey, you really got it right. Now, I suspect the person who's going to say you got it right does epidemiology. Just saying, okay. But it is sort of interesting to begin to use analytics, and the fact that we can now easily interrogate 3 million publications, that alone is kind of breathtaking, and we are doing more and more of this as time goes forward. Now, just to summarize this little piece of it, we clearly, NIH-funded UAB publications are outperforming typical publications. It seems like the more clinically focused stuff is really having an impact. And these are the 8 clinical areas where we seem to detect a very high influence, cumulative influence of what you're doing. So anyway, it'll be fascinating to hear what your faculty and your graduate students have to say about this. So let's talk a little bit about basic research. So when I was a beginning graduate student, I had finished my dental degree and I went off to graduate school. That was at a time, and I'm really going to date myself now, where if you wanted to obtain a restriction endonuclease, you purified it yourself. Okay, now I see some jaws dropping for those of you who think that all of those restriction endonucleases come from wherever you buy them now. We actually used to, in fact, I made enough Echo R1 for the entire University of Buffalo campus for 2 years with my own hands. Okay, very proud of that. Goodness knows how many patents I broke, but you know, there it is. Well, back then, when we were making our own endonucleases and we were pouring gels to sequence DNA, everybody's looking at me like I'm crazy, right? But you know, it's okay. We thought that if we could figure out the genome of our patient, everything would be solved. Everything would be solved. This guy's already shaking his head. No, you're stealing my thunder. And of course, we know that we still have a ways to go, although that sort of dream, if you will, you know, circa mid-70s, it's getting more and more real every day. And it's getting more and more real for all of these, you know, remarkable multidisciplinary, interdisciplinary projects led in part by NIH, but of course, the work is done by everybody around the world in the extramural community. And I'm sure each one of you can pick out one of your favorite projects in this space. Now, what's driving it? What's driving it is technology, because what was once, you know, $1 billion to do now is below $1,000, and people now are very upfront of saying we're going to see $100 sequence before too long. Now, that has a profound implication on healthcare, because if you can sequence a genome for $100, what a child is born is the first test that that child gets, their sequence. Think about that for a moment. I mean, you know, right now we test for certain metabolic diseases and conditions, but at $100 a test, so something to really think about. And when you are responsible for an enormous healthcare system, that has some profound implications for you, no doubt. So a really wonderful example of how basic science can turn everything on its head is the Microbiome Project. Now, I have to say, is there anybody here from the dental school? Yes, okay. Good. All right, I have to say to my physician colleagues, dentistry knew what the microbiome was 300 years ago. We called it dental plaque. Sorry, you guys are just catching up, okay? There, I said it. Feel yeah, right? I mean, you know. Okay, but the truth is, in order to really understand this remarkable superorganism, right? You know, you're born 100% human, you die 50% microbial, right? We needed a set of resources to characterize the microbiome. And so in phase 1, again, largely because sequencing became more affordable and more available, you know, over 14 terabytes of data became available, microbiomes from 300 individuals among different body sites. And now in phase 2, they're focusing on pregnancy and preterm birth, IBD, and prediabetes. And every day, every single day, there is another publication implicating the microbiome and something that you never, ever would have thought had anything to do with this. One of my very favorite, if you will, articles, which points to how profound this can be, is this paper that appeared in Science on how the intestinal microbiota influences cancer patient response to immunotherapy. The short of it is, when you look at responders and non-responders, it turns out that it's differences in the microbiome that could play a role. The implication being that if you have the wrong microbial composition, you could do a fecal transplant to reverse that, thereby enhancing the ability for the immunotherapy to work. It has some very profound implications. Now, because of the advent of inexpensive sequencing, putting a number of things together, the disorders with a known molecular basis is now approaching 6,500, but only 500 have therapy. So for those of you who are still in training and you're trying to figure out what you want to do with your careers, here it is. The gap between the 500 that we know what to do and the some 6,300, that gap, that's where the action's going to be in the coming years. Because we got to figure out what to do about all those other diseases. So it's remarkable that it's taken this long, but it's remarkable that we are now at the point where we do have a cure for the first molecular disease that was described. And I think you all know that sickle cell disease, it was described back in 1910. This is a prototypic disease that's in all the textbooks, but it's not a textbook. It's something that dramatically impacts people's lives who have this disease. And 1949, the inheritance was shown to be recessive, so it was understood to be genetic. 57, the basis was actually determined. 1980, so stop for a moment. The hemoglobin gene was cloned only in 1980, not that long ago. And look at how far we've come since 1980. The first approved drug for sickle cell, hydroxyurea, in large part by work on the campus at NIH. Recently, there have been bone marrow transplant successes, but the problem there, of course, is to have a successful match. And so today, using gene transfer with viral vectors, this seems to be the way forward. And with Cas9, genome editing as well. So this is work that is being done by John Tisdale in collaboration with Bluebird Bio. John's in our intramural program at NIH. I think you are familiar with this, where bone marrow is harvested and it is then transduced to rearrange the hemoglobin expression and then reinfused back into the affected individuals. And this has been now done on a number of patients, and it's working. It's really quite remarkable. And I hope, and if you haven't, Google it and take a look at the film clips. 60 Minutes ran an actually very good piece, a very measured piece about this, and focused on a particular remarkable patient following her progression in this regard. And of course, most recently now, we're thinking about how gene editing can be used for this, and there are a number of potential targets, as you might imagine. And of course, the technologies go beyond CRISPR-Cas9, but certainly that would be among the leading possibilities. So So the clinical trials, when do they begin? That's what everybody wants to know. Well, in fact, here's the announcement of it in clinicaltrials.gov, and it is recruiting, and and stay tuned. I mean, we'll see how this sorts out. But remember, this was the first genetic disease discovered, and we know we have treatments for about 500. We've got almost 6,000 more to go. Okay, now I'd like to change gears a bit and talk about how we support research in different ways to ensure that we get the maximum value out of the resources that we've been entrusted with. So let's start with rigor and reproducibility of science. So this is my most highly cited publication by far. I think my co-author may have something to do with that, but okay. But I'll take it, it's like. And so Francis and I put this commentary together on the NIH plans to enhance the reproducibility of science. And as I'm sure you all know, our application process has undergone some enhancement. And I'm sure some of you will let me know how well that's going when we meet later. It's okay, you can be candid. That's how we improve things, by getting feedback. But I want to focus particularly on an issue that we haven't spent a lot of time on, and it's going to get some of you crazy. Because otherwise, why should you be bored, right? So so how many of you use animals in your work? Okay, right, okay, so here we go. We're really concerned about underpowered animal studies. The UK is also. They've demanded much stronger statistics for their work. We have got to enhance the rigor of our research involving animals. There's all sorts of reasons why that are listed here. But it's sort of the equivalent of doing small, lousy human trials. Doing small, lousy animal trials doesn't get you anywhere either. And so among the things that we have found to be of concern, the power of the studies, or the lack thereof, the design, have you chosen the right species, does the model mean anything? One of the biggest challenges we have is that investigators create a model system back when they were postdocs, and they build their entire career on that model, whether it has anything to do with the human condition or not. But the whole field becomes so invested in the model that it becomes, you almost need an act of Congress to get rid of the model. If it's not the right model, we shouldn't be using it. Right now, overwhelmingly, the animal species that we use are mice and rats, but as you can see, other species are used as well. So this is just some data from circulation research, which just sort of surveyed many, many publications to see how many times the publication talked about that randomization of the animals. So if you use pigs, you're likely to do this. If you use mice, not so much, and everything in between. Sample size calculation. Now, maybe these folks did them, but they don't articulate that they did or how they did them. So if you use rabbits, not so great, and so forth. I mean, basically nobody reports sample size calculations anymore. People have called this power failure, kind of cute. It's a review in neuroscience reviews. And this really underscores the issue. So there are two standard maze tests that people use in biobehavioral work with rodents. One's called the water maze and the other's called the radial maze. And when you calculate the required number of animals for power at the 80% level, you need 134 animals per cell in the water maze and 68 animals for the radial maze. And if you want 95% power, it goes up to 220 and 112. The problem is when you survey the literature, here are the mean numbers of animals that are used. It's an order of magnitude too little. And then we wonder, we scratch our heads, we question ourselves, why isn't our work easily replicated? Because you're looking at noise. I mean, it's just that simple. And by the way, every one of us, how many of you have had a formal course in statistics? Well, that's not bad, because usually, although it is generational, people who look a little like me raise their hands very high. There is nothing holy about P.05. Let me repeat that. There is nothing holy. I mean, in fact, many people feel that what we really should be doing, if we're serious about rigor, is we should set it at 0.005. Now think back to the last few experiments you did. Okay, I mean, so the next topic, equally important, even more complicated, is addressing harassment in a research workplace. This has exploded over the last several years. I think in large part because of the courage of women who have come forward finally. And it's just very simple. We can't allow women to be driven out of science because of boorish behavior, or worse. The National Academies issued a report, Sexual Harassment of Women. If you have not yet read this report, please, this is absolutely required reading for every single member of the biomedical research community. Whether you are the president of the university or you are the technician in the lab and everybody in between. It is absolutely essential reading. The report describes 3 forms of sexual harassment. There's the unwanted sexual attention. That's the one that gets all the notoriety. The quid pro quo. But there is also gender harassment, which we don't pay enough attention to, which probably represents the vast majority of the issue, but it's the thing that we don't think about. It's the thing that we don't talk about. This is the sort of sexist hostility, the crude behaviors. These are the kinds of things that we've got to pay much, much more attention to. From the report, the cumulative effect of sexual harassment is a significant and costly loss of talent into the sciences, which has a consequence for the nation's economic and social well-being, not to mention the profound impact it has on the individuals who are affected. NIH has been trying to catch up because admittedly we had not been doing enough and are now trying to catch up. Francis issued a director's statement with a commitment to demonstrate our accountability and transparency. The statement also clarifies what we expect both institutions and investigators to do to ensure a safe workplace. If you are in a situation where there is harassment of any type going on, it's just not a productive, safe environment. And we can't expend the taxpayer funds in an environment where things are not safe. where things are not optimal. In the statement, there is a clear channel of communication to the NIH, and we are spending time now listening to victims and survivors of sexual harassment. And we are incorporating their perspectives into future actions. So instead of a bunch of old men talking to one another, figuring out what to do, which is usually what happens. You know, I'm one of those old men, so I can say that. You know, we're bringing in typically women, although not exclusively, who have been subject to this and have survived. We've also sent a letter to every funded institution clarifying what the expectations are. You've got to have a safe workplace. You've got to keep us informed. And we all collectively need to embrace our responsibility. There's a email address. Any concern of any type can be sent to granteeharassment@odnih.gov. We're not trying to short circuit whatever your university policies are. That's not the intent. And go through your university policies, and if they work, that's wonderful. Sadly, they don't always work at every institution. And there are individuals who feel they have no other recourse. This is how they can reach out. We have a website now and a communications campaign going on at NIH. We have administered a climate and harassment survey to our own staff, the first ever. We now have baseline data. We had about 60% response from our own employees, which is quite good. An interim report about this was presented a few weeks ago at our advisory committee to the director meeting. If you're interested in this, it's archived, it's on webcast. I would certainly recommend that you interrogate that. And then, as the penultimate thing that I'd like to discuss, the next generation of researchers. So I have some good news for some of you in the audience. My generation is eventually going to fade away. Broad smiles, which is great, right? I mean, because you are ready. If you would just get out of the way, right? Yeah. So I think all of you know what the issues are. We've got a workforce, the boomers particularly, who have aged, but actually the scientific workforce is older relative to the general population. There's some data here that you can see on your right. This is one of my favorite sets of data. It's sobering, but I think it tells the story. So if you look at the percent of funded investigators as a function of fiscal year, back in 1990, if you were, 45 or younger, you made up the bulk of the grantees. Over time, that group of people has gotten less and less, although we have finally leveled off and it's actually beginning to uptick a little bit. Mid-career benefited, but then leveled off and started to decline. But my group, my cohort, greater than 60, we're doing great. Now, what's wrong with this picture? Right? And so we've recognized this. We understand that this is not sustainable. And that if we don't reverse these curves, science is in trouble. Young scientists are not bashful. They are in constant pursuit of grant funding, and they don't have any time to do the science because they're always writing a grant. In extreme circumstances, some have even indicated that they have to cut corners. That scares me. Right? It should scare you. There is an overdependence on senior scientists. I know how wise we are, but you gotta let them go. It's like your kids. Throw them out of the house, they'll figure it out. Scientists report that some funding agencies are just driving them crazy with administrative burden. I don't know which funding agency they're talking about there. So the Congress took note of this, as you might imagine, and in the 21st Century Cures Act, they created the Investing in the Next Generation of Researchers. We also had an advisory committee to the director. Now, I don't know if you can actually see this photo, but what's really important about this photo is for the first time, we constituted a committee that had representatives from all stages of the career path. So in addition to a few gray hairs like myself, we actually had a number of young people, a graduate student, a couple of postdocs, assistant professors. Boy, did they let us have it. It was great. And this was such a valuable experience that going forward on every advisory committee to the director, we will ensure that we have people from all stages of career path. No longer will our groups just be gray-haired eminences. I think we'll be better for it as a result. In addition to this working group, there was also an Academy report. All of these things coalesced around very, very similar features. Here are some of the recommendations. Expand pathways for funding early-stage investigators through programs that do not require preliminary data. Why is that so important? Okay, you're in a lab, you're tolerating your mentor, you need a letter from him or her, you need preliminary data, you're beholden to them, they want you to do 10 more experiments before you leave, you can't say no, you're getting the picture, right? Some of you are living this now. Well, maybe not here, not at this institution, but at many other institutions. By not requiring preliminary data, you are liberating people so that they can come up with their best idea, because it may have nothing to do with the work that they trained with. That model that doesn't make any sense anymore, for example. NIH has accepted this recommendation. We're developing an R01 program. I think some of you knew the late Steve Katz, who was many years the director of the NIMS, died way too soon. And this award will be known as the Katz Award in his honor. Steve was very passionate about young investigators. I've told you about appointing scientists across career stages so that we get their perspective. And again, to sort of encourage the extramural community, as we are now doing at NIH, to look at how they're doing their hiring and their recruitment to ensure broad, diverse groups. Figuring out how we can stabilize the interstitial workforce, the so-called staff scientist positions, and encouraging people to come up with data that makes more transparent your own workforce metrics. So our approaches are working. Maybe not fast enough for some of you, but they are working. So we hit, this is the number of new investigators, early stage investigators, each fiscal year. And then as you can see with rescission back in 2013, who took it the worst? It was the early stage investigators. Ever since then, we've been really making a concerted effort to fund more and more early stage investigators by adjusting pay lines. Last year's target was 1,100. We funded well over 1,200. Our target for this year is 1,100. I think we're going to exceed it again. Now, this does come at the expense of something, right? It's a zero-sum game, but we think that this is the right priority to give things. Okay, so now the faculty can leave if you like. I'm now just talking to young people because it's like, I was actually kidding, but okay. This is like the best part. No, I'm okay. So it's really simple to build a research career. Here, here's the. That's actually real, by the way, in case anybody is wondering. And what I want to frame this is the top 10 things that I've learned throughout my career. Okay, but now I've got to explain to you who David Letterman is, because most, depending on the audience, I either start with Johnny Carson or I start with, yeah. So Google it. He was a late night host, and he used to do this thing about the top 10 of whatever it was that he was talking about. And so these are the top 10 things that I've learned from my career. The first is use experiments of nature to guide your research. Nature's a lot smarter than we are. Nature has figured it all out. We're trying to understand nature, not the reverse. So whenever you are sort of at that moment of, gee, now where do I go next? Let nature be your guide. Don't reinvent the wheel. The expert in the world is 3 labs from you. They're mean and scary, but knock on their door anyway and ask them for help. It's not a sign of weakness. It's okay to go for help. This is one that I tell everybody. Tell me what problems there are. I assume you're successful. Over-report failure, under-report success. Don't tell me how great you are. Tell me where things have failed so that I can help you. This is a very important one. You got to define what good is, and then you got to aim for it. And by definition, it's not necessarily what you are currently doing. Right? Because don't be complacent. Oh, well, what I'm doing is good. That's it. Now you're in stasis. You always want to aim higher. And the corollary to that is don't be afraid to fail. So when I interview people for positions, one of the first questions I ask them is, tell me about a couple of examples where you failed at something. And if they hem and haw and can't think of anything that they failed, I'm not going to hire them. Because it means they have never taken a risk. Right? It doesn't mean that they're geniuses. It means they just never took a risk. Failure's okay as long as you learn from it. Don't fall in love with your idea. By the way, boy, am I guilty of this one. Let your data be your compass. So humor can be an enormously effective tool, but only if the joke's on you. If you look around this room, you've got people from every place. And what is funny to a kid who grew up in Brooklyn may not be funny to somebody who grew up in India. It's just the way it is, okay? So if the joke's on you, if you're making fun of yourself, and for some of us it's very easy, I can do it without any problem, that's okay. But be careful about how you use humor. It can sometimes really backfire. Okay, again, and now I'm speaking from personal experience, hard work makes up for lack of brilliance. This is about perspiration, not about inspiration. Really. And don't have any doubt about that, please. Don't say, well, so-and-so is so brilliant, I could never do what so-and-so does. Baloney. You just got to work a little harder. Get up an hour earlier, stay an hour later, but you can make up for it. Most of you are here because you were always the smartest person in the room. And convincing young people that you don't have to be the smartest person in the room to succeed is a tough sell. You really don't have to be the smartest person in the room to succeed. But make sure you're always in a room with smart people. I've lived this for 19 years at NIH. To my left is Francis Collins. To my right is Tony Fauci. All I got to do is keep my dumb mouth shut, and I will learn something. So I've lived this, and it really, really matters, okay? So always be in a room with smart people. The number one and the most important, and I'll have to give you a little consecutive translation on this one, is be a mensch. A little Brooklyn is showing up here. It means to be a good person. It means to offer help without any expectation of return. And if everybody would do that and follow these other 9 top 10 things that I've learned, it's the pathway to success. With that, I want to thank you for your attention and happy to answer any questions that you may have. You're going to have to yell because my hearing is not great.
Regarding the animal experiments, cannabis, as we see that there is an issue with the reproducibility and transfer to the other species. So this is a common problem. So do you think that instead of using a single animal model, say for example C57 block 6 for a given experiment, can we mix up all the strains available and ask the same question and see if there is variation?
Right. So I think what you said, I hope what you said, was are there ways of combining experiments from different laboratories to enhance the power failure issue that I spoke about? And can we use more of a genetic outbred diversity to enhance what we're seeing, right? I mean, I think that was the gist of it. And the short answer is yes, that could be a very important way forward. Because it may not be, so let's say the power calculation shows that you really need to do 2,000 animals per cell. And that just may not be feasible for a typical R01 funded laboratory. But now what if we funded 10 laboratories, but the quid pro quo was you're all going to use the same protocol so that at the end we can combine those data. Now we are able to enhance the power of that study. Similarly, as I'm sure you know, based on the question you've asked, there are paradigms that one can use for outbred animals, which give you totally different results than the typical inbred, strains. So why do we, not do that more often? Well, because when we're trying to do genetics, it makes it more complicated. And if any of you have ever worked with brown Norwegian rats, they are miserable. I mean, they're not as docile and so on and so forth. So yes, both approaches are things that we need to incentivize and we need to enable. So if for example, we use the model where multiple labs are going to ultimately contribute to a common analytic, we have to provide the resources to enable that. So, but yes, that's the way that I think we can solve some of this problem. Yes. Now, you got to really yell at me. You got to yell at me like it's your wife or your... Huh? Okay, so truthfully, I didn't hear the question. So somebody in the middle, tell me what he said.
Was it the emphasis on early-stage investigators may create a discriminatory ban for early, mid-stage
Got it.
investigators who weigh in?
Thank you.
Was that it?
Yeah, good. Okay, thank you. So again, it's a zero-sum game. And it is possible that as one group is favored, another group is less favored. But in terms of the group that you are referring to, the mid-group, we are cognizant that the first competitive renewal is really a very important period in somebody's career path. And so we are actually looking very carefully at those individuals as well. Now, that doesn't mean that every single person who is, at the competitive renewal stage will be successful, but we are giving them special consideration for exactly the reasons that you're referring to. Again, early on we talked about should we be limiting funding beyond a certain threshold, and there were some members of the community that thought that was a great idea, and there were other members of the community that became extremely upset. Well, it's a zero-sum game. Personally, and then this is my personal opinion, I'd rather give an early-stage investigator his or her first award than give an established investigator their fifth or sixth R01. And that's just the way I feel about it. The individual institutes and centers have to do that calculus, and they do. Similarly, I'd rather give somebody their first competitive renewal, particularly if that means that without it, their lab shuts down, than to give a more established person, their fourth, fifth, or sixth R01. So it's the balancing act that one has to play. Well, yes. Maybe we do two more? Yeah, these two. So. Yeah, no, so thank you. So what about these early stage investigators? Will it be more difficult for them to get their second R01? And the answer is yes, it will be, because again, it's a zero-sum game. Our hope is that with awards like the MIRA Award that NIGMS is now using quite extensively, where the awards are a bit larger and a bit longer, that there will be at least a subset of investigators who won't need the second award, that the one award will be sufficient. But in some fields of science, that's just not practical. I understand that. But hopefully, with at least that first award, and with the understanding that the first competitive renewal will be looked at very carefully also, it will give the investigators enough momentum, that they'll be able to overcome. But you're right. I mean, again, it's a zero sum. With regard to individuals who, through normal, life issues, may need a little extra time or, perhaps a little different consideration, and there are a whole myriad of reasons why this may be true. The first stop is the program officer to have a conversation, because we do have some flexibilities that can be, taken advantage of. And we do have some programs which can, for example, provide additional support for technical help at times when an individual is not able to be 100% in the lab, again, because of various life occurrences. And so I would start with a discussion with your program officer, because they are knowledgeable about the things that, the latitudes that we do have. But yes, you're quite right. We do need to acknowledge that, and so start with the program officer. Yes, sir. I think you have the last word.
Thank you for your talk. I want to rephrase what my colleague asked, because I'm not so sure she was fully addressed. The thing is that, like you rightly said, postdocs get longer and longer.
Right.
There are different lengths of postdocs for different disciplines, which means that there are disparities in who an ESI
so you are really not an ESI, even though you are called an ESI, right? And so I guess one question is, how do you address that? And two, right now it seems like the window is gauged from after the PhD. I think it may be good to level the playing field if you make it time from when you get your position. It doesn't have to be 10 years,
Right.
maybe 5 years. But then the playing field is more level, and you're more sort of equal to someone who spent 2 years in the postdoc and someone who spent 8.
Right. No, thank you. So we debated vigorously the exact issue that you just raised. When should the clock start, and should we take into account the variable length of postdoc training? We came down on the sort of 10-year window, if you will, from the last terminal, degree, because we don't want to incentivize postdocs going too long. I appreciate that everybody's circumstance is unique and different, but in general, if you make it more advantageous to stay in your postdoc longer, you will have this sort of perverse result. Your postdoc will be longer. And ideally, what we would like to see is for individuals to shorten their postdoc time and to get into a more independent circumstance or a more permanent circumstance as soon as possible. But again, each individual's circumstance, is unique, and what would work, for one person may not work for the next person. We debated, is 10 years right? Is 12 years right? Should it be from the time of the appointment? I mean, we really did work through all of these, and at the end, we felt that the current approach make the most sense for the most people, but we understand that it will not, work for every single person, and unfortunately, the system just doesn't allow it to work for every single person. But anything that incentivizes the increase in postdoc training, we think would not be a good idea. So that's the reason. You may not like the answer, but that's the reason why we've done it this way.
Everybody, please join me in thanking Dr. Genome. And if we can, I'd like to present this very small and not over the limit recognition of your willingness to come speak to us. Dick and Gail, thank you for everything that you've done for us. And so, again, thanks everyone for coming.
Thank you. Thank you.